So we are on our way up, up again and again. Now we're going up to the cellular level, from atoms to molecules to cells. Now you may remember from that graphic where we talked about organelles, the mini organs that exist inside cells as well. But those are part of the cell, and that's what we're going to be talking about now, is the smallest living unit of the body. So what we're going to be doing in this chapter is going to be talking about the basics of how a cell works. What are the organelles? What's this plasma membrane that borders it? Remember that when we are looking at living things, they have these requirements, right, of life. Interactions with the environment, separation from inside and outside. And so the cell is going to have a lot of the same requirements that we have. And so you can see a little bit of yourself in the cell. Ah, yes. So we have to begin with a joke. And there's my cell joke. All right, so starting with the basics of the cell, this is our generic cell, right? We've got some sort of border on the outside, we've got this nucleus on the inside, and then we have these organelles. But there's a lot more details than that, so we're going to have to understand how the cell works, a basic cell. Because then when we get to our different organs of the body and organ systems, we're going to talk about the specialized cells of the body. So once we have the basic foundation of how a generic cell works, then we'll talk about how each of these unique cells in the body does its particular job according to its structure and function, or anatomy and physiology. Okay, so we start out with what's called cell theory, and that basically is, like we said, that the cell is the structural and functional unit of life, and the entire organism, like us, is going to be dependent on the activities of these cells. Like we said, as we get up to that level of talking about organ systems, cardiovascular system, urinary system, etc., we're always going to drill down to the cellular level in order to understand really how that system works. And like we said, we're going to see very unique structures of these cells, shapes of the cells, etc., so that they will give rise to their particular functions. So when we look at those functions through time, we can understand, again, how those tissues work, how those organs work, how those organ systems work. So we have this continuity of life that starts at this cellular basis, at the cellular level, and works its way up. The cells that we have in there arise from pre-existing cells, right? So you start out as one cell. Two gametes combined to form one cell, which was you. Then there were divisions that occur, mitotic divisions, to create more and more cells. And then once we had a whole army of cells, of kind of generic looking cells, those different cells could differentiate or specialize into particular cell types, like epithelial cells or muscle cells or nervous cells or connective tissue cells. So while there are only 4 basic tissue types in the body, and cells make up tissues, remember, there are over 200 different types of human cells. And again, they're very unique in terms of their structure and function. These just show you a few different types to show the diversity, even within a particular category, right? If we look at muscle cells, we have really, really long skeletal muscle cells that can be almost the length of an entire muscle, versus cardiac muscle cells or smooth muscle cells that are very small and very different in shape. We have different types of connective tissue cells. So you see fibroblasts here and erythrocytes. These are both connective tissues. Fat cells are also connective tissues. But notice when we look around the diversity of shape, we also have a diversity of functions as well, forming linings, transporting gases, storing nutrients, fighting disease, reproducing to make new individuals. Right? You see that cohesiveness of structure and function. Okay, so again, starting with the basics, the generalized cell. So if we talk about a generalized cell, we're going to have 3 basic parts, right? So we're going to have the plasma membrane, which is this flexible outer boundary. So, you know, when we were talking about a generic cell that you drew back in, I don't know, 1st grade, 2nd grade, something like that, you basically just do that, right? So you draw some sort of circle like this, and then You draw some sort of little dark part in it like that, and you're like, hey, that's a cell, or it's an eyeball or something like that. But this part that we have out here is the plasma membrane, the flexible outer boundary. Then we have the cytoplasm, which is all of this that we see in here, the intracellular fluid. Look at this word, intra is like the interior. This is within the cell fluid, containing the organelles, the mini organs that are sustaining the life of the cell. And then we have the nucleus, right? That's the dark dot that we drew right there. And that's where the DNA is, and that's the control center of the cell. Much like we talked about the brain and the spinal cord being the control center of you. So this diagram that we see here, quite a little bit different than maybe the one that I drew or that you drew previously. Lots of detail there. And you can see differences in terms of structures, and of course then differences in terms of functions of these different organelles that we see inside. Even the relatively simple looking plasma membrane that we see out here is going to be quite a bit different once we zoom down into it. Okay, so we said intracellular means inside, interior. Extracellular means outside, exterior. So substances found outside of cells, right? And those can be what we call extracellular fluids. So a lot of our fluids, think about a blood cell, right? It's got intracellular cytoplasm, but it floats around in the blood tissue. It floats around in a fluid that we call plasma. So that's an extracellular fluid, which is very important for the blood function overall. So we have extracellular fluids. This can be, for instance, I talked about plasma, but there's also, for instance, interstitial fluid. That's the plasma. There's also cerebrospinal fluid. We look at that, right? Cerebro makes you think of cerebrum, brain, etc., and around the spine. So there's a fluid that goes around the brain and the spine that bathes the nervous system. So these are different types of extracellular fluids that are important. And the reason they're important is, remember, living things are going to interact with their surrounding environment. And so that's what we're talking about. When they're exchanging things across their plasma membrane, they're exchanging it with the extracellular fluid. Now there are also some cellular secretions like saliva and mucus, etc. And so when they're inside the cell, that's intracellular fluid, but once they release from the cell, then they are extracellular fluid instead. Other things that we find outside of cells, extracellular matrix. This is important in terms of a tissue, right? When we have cells that are working together and often associated or stuck together, it's this extracellular matrix like glue which adheres them to one another. Okay, so what we're going to do is we're going to take our tour of the cell. We're going to start with the plasma membrane towards the outside. We're going to work our way towards the inside. So a plasma membrane, in terms of what it does, is it separates the inside, the intracellular fluid, or ICF, from the outside, or the extracellular fluid that we would call the ECF, abbreviate, right? Now when we look at it though, even though we drew it as a very simple circle like this, right? In fact, it's not a wall, right? We're not building a wall like nothing's going to get across, nothing's going to get inside, nothing's going to get outside. It is more of a border where, and in fact, you know, you talk about borders and walls these days, you can get into all kinds of other topics, but realize that the border for a cell is meant to control or regulate movement across it. And so most borders are in fact like that. It's to control what is coming in and what's going out, but it's not to totally separate and not allow any movement at all. It's very important to control and regulate the movement that is going across it. Okay, so plasma or cell membrane. When we look at it, the structure of it has a lot of different molecules to it actually, mostly lipids organized in a bilayer. A bicycle has how many tires? 2 tires. A bilayer has 2 layers to it as well. So a lot of lipids in it that we're going to look at, but there's also proteins in it as well. So that's another type of molecule. And so since there's these different types of molecules inside of the plasma membrane, we typically call it a fluid mosaic. So the fluid aspect of it means that it moves. So these different individual lipids and proteins move relative to each other. And then it's called a mosaic, like you think about a mosaic piece of art. It's made up of individual pieces that then, when you step back, make a larger form. And so all of these individual molecules of different types make up the whole of the plasma membrane. So lipids, proteins, sugars. You see that there's all kinds of different molecules that help to make it up. And in fact, there's also oftentimes structures that are within the plasma membrane that will help to, excuse me, adhere cells to each other, forming cell junctions. So this diagram that you see here zooms up on that plasma membrane and really shows us the detail of what we're talking about in terms of this fluid mosaic. So again, here we see some of our lipids. So here is one of our phospholipids that makes up most of the plasma membrane. And you can see it's organized into a bilayer. So here's another one extending across this way. So you can see a bunch of polar heads extending one way and nonpolar tails extending this way. And then here's some other molecules that you see here. They kind of look like little jellyfish if you're a marine biologist. Right? So lipid bilayer, 2 layers of those lipids, and then we see proteins that extend all the way through. They're found only on one side. We can also see some of these sugars attached to it. You can see cholesterol even embedded within it. And you're like, ooh, cholesterol is bad. But in fact, it helps to stabilize the plasma membrane as well. So it has a very important function, like we said, of separating the inside of the cell from the outside, but also controlling the movement and the relationship between the inside and the outside, getting what we need inside the cell, getting rid of waste products, etc. Also, it can allow, if you will, communication or relationships with other cells, etc. Right? You can have molecules that are on the outside of it that are like ID tags that identify it so it's not attacked. You can have different types of receptors that bind chemicals like hormones and neurotransmitters, etc. for communication. So lots of different functions like we see there. So lipid bilayer made up of mostly phospholipids, about 75%, where, like we said, they have that very unique shape to them where we have something like this, right? We have a head to it with 2 tails coming off of it. These are the fatty acid tails, which are nonpolar, and these are called hydrophobic, right? Water hating or water fearing, actually. So that part of it does not get along with it. And those tails are extended towards the inside of the plasma membrane. So we have in the extracellular fluid out here, that's mostly water. The intracellular fluid here is also water-based, so it makes sense that the polar heads that get along with water are facing those 2 aqueous environments, and the nonpolar interior helps to control that movement of different types of polar molecules through it. So hydrophilic or water-loving on the outside, hydrophobic on the inside. Kind of like an Oreo cookie, right? You got the fat in the inside there, right? Okay, so mostly phospholipids. You do have some glycolipids as well that dot the outer surface that can be used as ID tags, etc. 20% cholesterol, like we said, for stability. And then also, if you look at that, this is just looking at the lipid bilayer itself, but we know that there are other types of molecules in there too, in terms of, like we said, sugars and proteins, etc. So the proteins have lots of different functions. Again, you probably saw on that graphic, different forms, different structures give different functions. So they can be allowed to communicate. They actually have a lot of different functions. They can allow passage, safe passage through the plasma membrane. They can bind to particular substances, etc. So some of them are attached, some float freely in that fluid mosaic. So they are very, if you will, varied in terms of their structure and function. Those that are attached are called integral proteins as opposed to peripheral proteins. Integral, right, kind of centrally, firmly inserted into the membrane, as opposed to peripheral, are kind of at the edge. So integral proteins are going to basically go, for the most part, through all the way through the plasma membrane, and oftentimes they will transport things like as channels through the plasma membrane. Some of them also act as enzymes. Remember what enzymes do when we were talking about chemistry? Enzymes are proteins that are acting like catalysts. So they are helping with chemical reactions. Or receptors, again, kind of like the hearing part, the ears of the cell. The peripheral proteins also can act like enzymes. They can also act as motor proteins to change the shape, or even adhere cells to each other instead. So this figure, like we see here, shows you a variety of different protein shapes and different protein functions. So do check it out. It's figure 3.4 in your textbook. All right, we also said that there's sugar sticking out of the plasma membrane. These ones tend to give us, if you will, a different sugar coating on each different type of cell, and that helps to identify that particular type of cell for cell-to-cell recognition, so that like our immune system doesn't attack it, or that we can see, the body can see that those cells are part of one cohesive tissue. Now what we find is, a small aside, some cancerous cells actually change that sugar coating so rapidly that whereas one time our immune system cells would be able to hunt down and recognize those cells that are different from our own, the changing sugar coating, even if they start to recognize it or recognize it one time, now it's different than what it was before. It's like changing mass on the outside of the cell. So that makes it difficult sometimes for our immune system, our bodily defenses, to root out, if you will, all the cancerous cells. Okay, so we said that cells often contribute to tissues. Tissues are where these cells are working together for a particular function, and they're often adhered to one another. So some cells are free, like blood cells and sperm cells, but most of them are bound together. And there are three ways that we can bind these cells together. Kind of like chemical bonds, like we talked about before, but those are atoms creating molecules. And now we're talking about cells creating tissues. So, three different ways that cells can be bound: tight junctions, desmosomes, and gap junctions. So tight junctions, as you can imagine, as the name indicates, these really seal these two cells together. They're impermeable. Things cannot get through the tight junctions. And so these prevent things from slipping in between cells. So for instance, if you think about your shirt, you've got different types of seams where it is sewn together, and so things can't get through those areas. Even though it's two different pieces of cloth, you can't get anything going through there. So that's like a tight junction. Desmosomes is where we're anchoring cells together, but at a particular spot. It's a little bit like Velcro, and I'm sorry I don't have a Velcro shirt to really demonstrate that. I should, otherwise I'd be kind of ripping it apart, and that would be distracting. But what they do is, at a particular part, they're going to adhere these cells together to each other. So desmosomes are a little bit like Velcro, where we're holding them together. You can almost think of it like a button as opposed to Velcro, right? So if you see a button, these two parts of my shirt are held together in that one spot. But adjacent to it, you can actually get passage of materials. So desmosomes are little spots, spot welding, or a button or a Velcro in one particular spot. Then we also have gap junctions. Now gap junctions are very, very specialized junctions made out of proteins that do hold cells together, but notice that there's a channel passing through the center of it that allows the movement of particles like ions to move directly from one cell to another. So very important in like cardiac muscle, etc. Okay, so we talked about the different types of molecules that make up the plasma membrane, mostly that lipid bilayer from the phospholipids, but other types of lipids as well, like cholesterol, etc. Lots of different types of proteins and things like that. And we say that the function of the plasma membrane is to regulate the movement of substances across the membrane. So these plasma membranes are what are called selectively permeable. That means certain things can get through, right? They're not freely permeable. Certain things can get through at certain times, often associated with those proteins. So two ways that the substances can cross a membrane: a passive process, which doesn't require energy, and then an active process. So if you're passive, like if you're with a group of friends and you're trying to decide what movie to see, you're like, whatever, I don't care, I'll just go with the flow, right? No energy in terms of making the decision. That's passive. You're just going to go with everybody else. Active processes, you're like, well, I don't know, let's check Rotten Tomatoes, and I don't want to see this, and I want to see that. You put a lot of energy into the decision. Active processes require our energy molecule ATP, and so active processes are going to actively push things across the membrane instead. Okay, so let's first look at passive membrane transport. So we have two different types. We have diffusion, and then we also have filtration. So diffusion actually has three subtypes, if you will. Simple diffusion, carrier or channel mediated facilitated diffusion, and osmosis. So simple diffusion, it's like if you put the Kool-Aid into the water and you see those particles just kind of spread out, right? Mostly it has to do with concentration, right? So high concentration in one area, and we're going to be spreading out from high concentration to low concentration, from high to low, like a ball rolling downhill. So a ball rolling downhill doesn't take much energy, right? It basically just naturally goes from high to low. So that's kind of like simple diffusion. Now carrier or channel mediated facilitated diffusion, to facilitate something means it needs some help. And so we may have a concentration gradient. We may have like a high amount of glucose outside of the cell, and we want it inside the cell, but it can't get across that plasma membrane. It needs a carrier, a protein, to assist it and escort it across. So that's a type of diffusion as well. And then there's osmosis. Now you guys know about osmosis. I'm not going to tell you about that, right? That's how you study, right? You put your head on the book and you just hope, right? But you're looking at a concentration of knowledge from the book, hopefully going into your brain from high concentration to low concentration. But osmosis is not, according to knowledge, it is water, right? So the diffusion of water from an area of high concentration of water to an area of lower concentration of water is osmosis. Again, another passive process. Filtration is passive as well. It doesn't take ATP, but usually there is fluid pressure pushing it across, like making coffee, right? So you pour water in there and it pushes through the filter because there is the mass of the water pushing, exerting in a force and causing it to go through the filter itself. Okay, so this is just more details about those different types of passive processes. Like we said, diffusion, we're going from an area of high concentration to low concentration or less concentration, and so like a ball rolling downhill. And so it's going to be influenced by the size of the molecule, just like the size of the ball, and temperature. So temperature is energy, right? And so if there's more energy and things are bouncing off each other more, then we're going to tend to move a bit more as opposed to if things are slowed down. So this just kind of shows high concentration moving to low concentration, kind of radiating out, right? Until equilibrium is reached, evenly distributed. Then there's still going to be some random movements, but it's not going to be all of this kind of directed movement like we see here, away from high concentration going to low concentration. So it's kind of like, you know, if you're at a party or something like that, you're like, oh, it's getting kind of crowded in here, and you know, people are spilling their drinks on you and stuff like that, and you look into another room in the house, you're like, hey, there's only a few people over in that room. I think I'm going to migrate from high concentration here to low concentration there, where I have a bit more elbow room. So molecules and ions are like that too. Okay, so diffusion, like we said, there is simple diffusion, but often that plasma membrane, the structure of it, blocks a lot of the diffusion, kind of the easy movement across the plasma membrane, and so usually we need some sort of escorting across. So carrier molecules that are going to escort larger molecules, or basically if we look at different types of, if we talk about polar molecules or something like that, they're not going to be able to get through that nonpolar core that we see in the lipid bilayer. So some small molecules would be able to get across, like water, etc., but other ones, bigger, nonpolar ones, etc., lipid insoluble ones, have to be escorted via the protein like we see there. There's some of our channels, osmosis, right, the movement of water. If we look at these Mickey Mouse, dead mouse molecules like we see over here, high concentration, low concentration. So they're gonna want to move from high to low like we see there. So it could be through channels, protein channels, or it could just be through the lipid bilayer. Now what we find is that, you know, in a particular solution, remember a solution is going to consist of solute and solvent, oftentimes water. And so water is going to diffuse according to its gradient, and the solute is going to diffuse according to its gradient. So when we have a permeable membrane there where these can kind of move from one place to another, then both of those molecules or ions are going to be moving according to their own gradients. But sometimes if you have a selectively permeable membrane, what you'll find is say water can move through the membrane and this particular solute can't, we'll see the water move. The sugar may want to move, but it can't get through. In this case, due to the size of the pores that we have here, but the water can, so we'll actually see the water diffuse according to its gradient, right? There's more water here. If we look at the concentration, right, we see a lot more blue space here compared to over here. So there's a higher concentration of water here relative to the numbers of solute molecules compared to over here. So the water is going to want to go this way. So when the water moves, that creates pressure, right? Because we're packing more and more substance into this side over here. So that actually is going to create what we call osmotic pressure. As the water moves into a cell via osmosis, that's going to create osmotic pressure. There's also hydrostatic pressure, which is from the outside of the cell, right? Hydro means water. And so these pressures are very important in terms of maintaining cell shape and also in terms of fluid systems like the urinary system. Now, like we said, in terms of cell shape, when we have fluids moving into the cell and out of the cell, cells kind of drying out and then filling up again, that can change the overall volume of the cell. And so it's very important for our cells to be surrounded by a solution, an extracellular fluid that should be what we call isotonic, right? Iso means the same, like an isosceles triangle. So it should have the same osmolarity or concentration inside and outside the cell, so that the volume doesn't fluctuate, doesn't go up like a balloon and then shrink down, etc. In a hypertonic solution, what happens is, hyper means a lot, right? What happens is a hypertonic solution has a higher concentration or osmolarity compared to the cell. So that means that we have really concentrated solution here, not a lot of water and a lot of that solute, and inside of the cell we have a little solute and a lot of water, right? So the water is going to want to diffuse from high water concentration here to there, where it's a low water concentration. So the cell is going to lose water and it's going to shrink when it's sitting in a very concentrated hypertonic solution. In a hypotonic solution, it's going to do the opposite, right? Hypo means low, and so in a very dilute extracellular fluid, the water is going to go from high out here to inside the cell, and it's going to tend to blow the cell up instead. So you can see that here. Isotonic solution, small exchanges across the membrane. Hypertonic solution, where we're losing water to the surrounding environment. And then hypotonic solution, where the water is going into the cell instead, causing it to swell up and possibly lyse or even explode. That's why when you go to the fish store and you see all those saltwater fish, you're like, ooh, those are pretty ones. I'm going to go home and put it in my freshwater tank. You can't do that, because saltwater fish are adapted to living in a higher concentration solution. And so you put them in a freshwater solution, this is what's going to happen. You're going to put them in a hypotonic solution relative to their cell concentrations, and all their cells are going to swell up and they're going to pop. So you put your ear to the aquarium and you can actually hear all their cells popping as they die. No, just kidding. But don't do that. Freshwater, saltwater, don't mix. Okay. So right now what we've done is we've talked about the beginnings of the cell. We've defined the cell. We've talked about the unique and detailed aspect of the plasma membrane. We've talked about movement across the plasma membrane that doesn't require energy. And now on the horizon, we're going to talk about that energy requiring transport called active transport. But we're going to stop right here. It's kind of like a cliffhanger, and we'll pick up that on the next lecture instead.